Good afternoon, everyone. Welcome to the next lecture of Professor Martin Bucher. And I just wanted to make just two reminders. One, as I mentioned earlier this morning, this afternoon at 4, there is a NITEP colloquium in which Ilya will speak about open quantum walks, which is something closely related to the theory of open quantum system and their simulation, but not something that he will cover in the formal course. So if you're curious to see something a little bit different, please join us this afternoon at 4. I shared the link in the previous Zoom call, but it's also in Moodle. So I think so. Yeah, you should find it there. Yeah. If not, just let me know. And the other thing is, don't forget our ongoing competition for the best question of the session. And I see there already in the chat people complaining that I haven't received the voucher yet. We will issue the vouchers only at the end of the school because we will do them all in one go. Otherwise, if you have every day to go and get 2 vouchers, become a little bit painful. Yeah? So be patient. We made a note of all the winners. And after the school, we will distribute all the vouchers in one go. So don't worry, just be patient for another 4 or 5 days. Yeah. So Martin, sorry, this was just the bureaucracy at the beginning of the talk. Now we are all looking forward to see how your story continues and unfolds.
Okay.
You're welcome to share your screen.
Yeah. Let me do that. And let's see. I'll go here. Let's see.
Perfect.
Oh, okay. Okay, so I'm going to continue from where I left off last time, and this lecture is going to be a little more molecular biology, biotechnology, and then in the next 2 lectures, I'm going to talk a little more about maths and modeling and statistical mechanics and algorithms. But in order to appreciate the problems, I think you need to know a little about what's going on and how people can probe genomes and do experiments, not necessarily all the gritty details. So this is sort of a bare bones what I think is important to know. I also put my one of my email addresses in case you want to write to me, and I'm also curious for those who have done the program that I suggested, what results you have obtained. So let me maybe start by asking you whether there are any questions from the previous lecture or the previous 2 lectures. Let's see, I can't really see the screen, and so we could take some questions now and at the end of course. Okay. Well, let's go back to DNA and RNA only differs by a little, and as we shall discuss. And so this is the famous quote from the first paper of Watson and Crick, where their contribution was deducing using X-ray crystallography and other considerations, this double helix structure. So they write, it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism of the genetic material. So, of course, they're right here, and when we look at this double helix, one of the important points to note is that the bonds between the two bases are what are so-called hydrogen bonds, and the feature of these bonds that is important is that these are weak bonds that can easily be broken, as opposed to the rest of the bonds, that hold the bases together and hold the backbone together, which are covalent bonds. And when we speak about weak or strong, it's relative to the thermal energy, so K Boltzmann T. So a single hydrogen bond, that's what makes water not be a gas like methane, because the molecules stick together more than one would otherwise expect, with van der Waals forces and stuff like that. But it's not enough to make ice, at sort of normal temperatures. So we'll talk, so that this sort of makes the structure a bit like a zipper, which is important. So here is sort of schematic diagram of the copying mechanism. And you can see this is sort of like a machine where you have the double strand being fed in from the right-hand side. And then it is split into, it is pulled apart. And then you have these different bases attached onto a backbone fragment that sort of come into place because of the hydrogen bonding. And then enzyme, the DNA polymerase, sort of puts the different, snaps them together. And this sort of moves on at a rather fast rate. And there's also error correction, so that in the case of DNA, this happens with very few errors. But there are errors which are mutations that occur in the copying process. And that's going to be one of the things that we're very interested in a lot of, well, for a lot of reasons. It's a useful marker for ancestry, for all kinds of things, and it tries out new combinations. Another detail here that is somewhat relevant is that these strands, you see them sort of labeled 3' and 5', and those are sort of the two ends. And these polymerases are actually not the same on the two sides, because they one has to add on the adding to the 3' side and one through the 5' side, and you'll see that that's important in the polymerase chain reaction, which is sort of one of the tools that is useful for all sorts of things. So RNA can be copied directly, onto RNA, but it is, a lot more error prone and creates a lot more mutations. So the coronavirus is an RNA virus. Okay, so I think this is, here you see more in terms of bonds, what is going on in the sort of blue, green, and red and purple. Those are the bases which are different. So they have A, AT, G, C, and AT is always matched together, and G, C is always matched together. And you sort of see, if you go to, let's see, the oxygen bridge, and you start counting, where the base group gets added on. This is the first carbon, this is the second carbon, this is the third carbon. So that's why this is called the 3' end. And then 4, 5, and then the phosphate. And then this is, going to be removed and bridge onto here. And this is what holds the strands together, but they run in opposite directions. So this is something that will you'll see is important later on. Okay. Yeah, these are the bases, and I think you can sort of think of this as a bit of 3D printer that you have 4 types of dye rather than 3, and they're 3 colors, and the rest of the stuff that we were looking at are just catalysts, and that put these different parts together. So these are the consumables for making copies. So in a cell, there's a replication of the DNA strands. So you could have in simple bacteria or viruses a single strand. Sometimes they're linear, sometimes they're circular, but in a lot of organisms, you have several strands. So, in the human, there are 23. The influenza virus has has 8 strands, so there, that is a bit similar to a chromosome. But in you know, higher organisms, you sort of want to reproduce so that if you have n chromosomes, then you you replicate in the 8 different types, or you know, in each corner, so you don't have 2 of one type, or I mean 3 of one type, or just 1, or something missing. There has to be this matching. So here you see, first there is the replication that takes place, and the 2 strands are held together. Another thing we'll see is that it's not just the case that you can, you know, you add this enzyme and it just spontaneously everywhere and anywhere going to duplicate. So there's sort of a center of duplication where it starts, and in the you know, case of the higher organisms, it's sort of held together. So this is the outcome of just the DNA replication. And then there are these microtubules that form and segregate one of each kind by pulling, you know, these centromeres, the 2 identical halves get pulled apart, and then the cell can divide. So in terms of, you know, in terms of genetics, one of the important things is going to be whether there you have organism where they're all you have N chromosomes that are all different from each other. So they all carry genes, and that's called a haploid organism. So haploid is derived from the Greek word simple, and then diploid is where they're of each type, there are 2 copies of the gene, so they're so-called homologous pairs of chromosomes. And we're going to see that there are significant advantages to having these pairs, because it allows new combinations to be tried out in the, you know, absence of mutations. So if you would take our sort of simplest example of a haploid organism, which would be something like the coronavirus or most bacteria. I mean, there's more to the story. You just have the same thing gets reproduced with some errors. There's going to be some natural selection, but basically all of the mutations that are present in terms of selection, it's kind of take it or leave it. And you can't sort of try out, well, what are the good new mutations, and what happens if you combine the good ones and let the bad ones aside. It's sort of I'll take it or leave it. And there's quite a bit of mathematical theory that is not necessarily the best thing, you know, whereas when they're pairs of chromosomes, you can sort of try out different combinations, and there's a lot of genetic diversity that is possible even without mutation. So just the genetic diversity that exists, different types of genes, that can be sort of shared in subsequent generations, and it allows organisms to adapt faster to change. So we will explore that somewhat in mathematical models. Okay, so this is just, except for mistakes, this is, copying identically and creating a pair of cells that are theoretically exactly the same. And then if, something goes wrong, the cells can be killed off, but that isn't such a big deal. So here we go on to the sort of sexual reproduction, where there's sort of a splitting of the chromosomes. So before you go to a situation where you have 2 copies of each chromosome, and then there are 2 duplications here that take place. But in the end, you get 4 copies that have, a different assortment of, is it, taken from the chromosome of the father or the mother? And this is what allows there to be genetic diversity without mutation. And so new combinations get tried out very, very quickly. The next generation is not just, the mother take it or leave it, or the father take it or leave it, but it's some sort of combination. And we'll see the number of combinations is really immense. Okay, so there's also this phenomenon of crossing over, which means that it's not just, say for chromosome 1, that you take the chromosome of, either the father or the mother, that, you can take part of one and then they sort of cross and swap ends, and that can happen more than once, and that can happen at many different places. So this is something we discussed last time with the genetic mapping with correlations that were observed that violated the rules of heredity as established by Gregor Mendel. So Thomas Hunt Morgan explored these correlations very quantitatively, and he deduced just on statistics a sort of linear map of the genes. And so that's because of this process that takes place here. Okay, so let's, oops, this should be 2 to the 22. Okay, so sex chromosomes are a little special, so we can talk about that in a second. But if we just look at the 22, autosomal means basically a non-sex chromosome, where the 2 parents that are contributing provide the same set of genes. So if we didn't have any crossover, then we would be making a binary choice, 2 to the 22 times. So 2 to the 10 is 1,000. So we get 4 million different possibilities. So that would mean that, you know, a couple would have to have 4 million children before they would, you know, under these rules, that they would have 2 children who are genetically identical. You know, that's barring identical twins. So you can see that there's a lot of scrambling up that occurs. But because of crossing over, and crossing over occurs about, you know, very roughly once per chromosome, you get even more diversity. So you have this process where if we you know, go through many, many generations, there basically is no correlation between, you know, having one type of gene and having at one site and another at another site, that they're like random, statistically independent choices. And, you know, that requires having crossovers occurring between those 2 locations. So I don't think it's, you know, that necessary to, you know, be too quantitative about this. But you know, if there are 2 choices for each gene, which is just a rough estimate, then it would be 2 times, you know, the number of distinct genes. Yeah, so I mentioned that there have been a lot of theoretical, mathematical articles written why this sort of mode of reproduction is advantageous. And we will discuss this in future lectures. Okay, so this will become important when we talk about phylogeny, making, you know, family trees. You know, it could be for persons, or it could be, you know, for the coronavirus. So if we try to do the same thing for people, well, if we just look at male descendants, then the Y chromosome is, you know, unique to males. So there isn't going to be this this mixing, and that sort of allows trees to be established, which is, you know, very useful for some things. It's also sort of interesting in that the handing down of sort of male genes is more like bacteria and viruses rather than the, you know, sexual gene selection. So there, when we're talking about this, you know, diploid reproduction, it's really a little separate from sexual differentiation, and it's not necessarily unique to, you know, very, well, all higher organisms, you know, all vertebrates, or diploid. So that would be sort of a clue that have a more complicated organism with a large degree of differentiation of cells. The, you know, diploid method is preferred, but yeast, for example, can both go through a haploid and diploid phase, and so they're very simple organisms where the reproduction takes place in this way. Okay, here is the crossing over, and when we look at this the diagram of meiosis, I think this was the second stage. You had these, you know, two sets of copies, and they can, you know, cross segments, so that everything was either blue or sort of hot pink here, and then there was a swap made here. So this is what is known as, you know, recombination. So there, you know, new elements and new combinations that are being brought in. So this is sort of physically how it happens. And it turns out that, you know, rather than probabilities are sort of these hot spots, that there are a lot of them where there are these breaks that occur and then they get fixed, that two ends might not get reconnected at the same position if they both break in the same place. So that's sort of physically what is happening. But if you have a large number of hotspots, it sort of seems like a, you know, almost Poissonian process where you might have a sort of mapping of physical distance in terms of base pairs, and, you know, what is the genetic distance, which determines the probability of crossover occurring. Okay, so viruses are not to be left behind, and even though, so this is the influenza virus, and we see that, well, there are 8 different strands here, and each of these, you know, duplicate, and so this is the viral envelope, and so someone has the flu, it invades the host cell, it uses some of the machinery of the host cell to, you know, replicate its RNA here, make the proteins for, you know, new virons, and then assembles them together. And if a person just has one type of flu and doesn't have 2 flus at the same time, then, you know, if everything goes well, you're just going to have a copy of this, a copy of that, a copy of that. But you can also have these events, and that's why the different strains of the flu, they're, you know, H some number, N some number, those refer to some of these proteins that they can get swapped in different combinations. So if there are 2 to choose from, then you have, you know, 256 possibilities for the next strain. And you see that happens with some of the more serious new flus that happen. And they're more than, I mean, if there were just mutations and these were just fixed, no mutations and fixed motifs, then you would have, from this mechanism of reassortment, 256 possibilities. But of course, there are mutations that happen along these different strands of the RNA, and that allows basically an endless number of combinations. So this is somewhat similar. And I mean, recombination is sort of a bit of a buzzword. It sort of means anything sort of unusual that happens where new genetic material is brought in, you know, to create to create a new genome. So haploid organisms, or organisms that are always haploid, they don't have this sort of like yeast, you know, 2 stages. They can either reproduce sexually or asexually. There are other ways of bringing in new genetic material. So let's talk a little about viruses. And this is actually, you know, most classification systems are rather boring because it's, you know, just trying to put things into nice boxes. But viruses are, you know, much more interesting in how they replicate, and because there's in bacteria and higher organisms, it's always, you know, DNA carries the information, and then DNA gets, the genes get transcribed into messenger RNA, and then the messenger RNA goes to the ribosomes, and it's turned into protein. So it's all sort of, you know, DNA on the top, and here, you know, with 2 strands. So the different classes basically of viruses have to do with how the replication takes place. So there are viruses that are double-strand DNA. So that is, you know, roughly the same sort of standard scheme that we just discussed. The only thing that makes them a virus is that they cannot reproduce on their own. They need to infect a host organism because they need to use some of the machinery of the host organism, for example, to make their proteins that are coded in the DNA. But this is one of the options. So this is what David Baltimore calls class 1. Then there's, so the thing at the top is sort of what becomes encapsulated, you know, in this little shell or the capsid that, you know, goes out and infects other host cells. Then SS is single-stranded. So first it has to become double-stranded. And here at the bottom, this is a messenger RNA transcript, and then the body of the, well, the host cell that has been infected will change this into the viral proteins to make more virus, et cetera. Okay, so you can have double-stranded RNA, and that is sort of interesting because that means that with these RNA viruses, they need to provide their own enzymes to replicate the RNA, because that's not part of the mechanism of any of the host cells. that are going to be more like the class 1 virus. So that's going to be one of the proteins that is encoded in these RNA viruses, and basically almost all of them. Then there are two classes of single-stranded viruses in the RNA viruses, and one is called positive sense and one is negative sense. So the coronavirus is in this category, and the influenza virus is in the negative sense category. So positive sense means that the virus can create its own proteins without going into the nucleus. So the viral genome can just use the apparatus to translate, the segment of its RNA. It just acts as messenger RNA. Whereas in the case of the negative sense viruses, they basically need to use the mechanism in the nucleus of the cell to, create a complementary strand of messenger RNA, which in turn makes their proteins. Then we have the retroviruses. So RT is retroviruses, where the RNA gets turned into DNA, and then that can be incorporated into the genome of the host cell. So that's one of the things the HIV virus does. In that, once you're infected, you can't just kill off all of the virus. That wouldn't suffice, because it has been, at least in some of the host cells, incorporated into the DNA. So, that's also one of the reasons why the infections are so long, last for such a long time. So something like the coronavirus or the flu virus, the strategy is basically, you allow the host cell to survive for as long as you need to, make all the proteins and all the replication of the RNA to make many, many virons, but then they want to get out and infect other cells. So there are proteins that make the cell just burst and die, and all these virons spread all over the place. Whereas with these retroviruses, that can happen, but it doesn't necessarily always happen. Instead, there's sort of more of a long-term strategy of, taking root in the DNA and being in a sort of latent state to be able to break out later on. And then finally, there are these other viruses that change into DNA, RNA, and different. There's sort of a switching back and forth. So this is a lot richer set of combinations than you have in the higher organisms or all of the host organisms. Okay, so I'm just going to mention, and this is just one of the possibilities that, so if we have organisms that are not diploid, and then there's sort of the desirability or necessity of acquiring new characteristics. And plasmids are basically small pieces of extracellular DNA that reproduce separately of the chromosomes, and they can sort of provide to bacteria interesting new abilities. So for example, antibiotic resistance would be one of these. And here we see that these plasmids, which are rather small, that they can be exchanged between bacteria, both of the same species and sometimes of other species. So there's this structure called the pilus, and there's this bridge that forms, and then there's a replication with a break, and the plasmid DNA goes over to the other bacterium. So now, if this is antibiotic resistance, the bacteria has acquired this, this new ability. And there can also be exchange of segments of DNA between the chromosome and the plasmid. And it's also sometimes happens that stray segments of DNA can go through the cell membrane and become part. So this is sort of not the usual thing that happens, but this is a way of bacteria sort of and other haploid organisms, or always haploid organisms, acquiring new genetic material. And I won't talk about it, but this can also, these sorts of mechanisms can also be used to add genes and are useful for genetic engineering. Okay, let's sort of look at orders of magnitude a bit. So I will let you go through these tables on your own, but it's interesting to look at genome size because each base pair is 2 bits, so that's sort of the number of base pairs is, in a sense, the entropy or the amount of information to create an organism. I mean, there might be a lot of sort of trash information included as well, but so not all of it is necessarily relevant, but this sort of places an upper bound on the complexity. And the sort of shortest virus is 2,000 bases. So here it's not bytes, it's bases. So 2 to this power, 4 to this power is the number of combinations. I mean, they're base pairs in many cases because there's a complementary strand, but that carries exactly the same information, because it's just, you know, changing A to T and C to G, etc., and vice versa. And the coronavirus is 30,000, so it's sort of on the biggish side. And then we got 2.747 megabase pairs. This is sort of the record. Then let's see. I should say, I'll go on to the, you know, bacteria, but they're bigger, and but not that much bigger than the viruses. So here you can sort of see the range. So the mitochondrion is there's human DNA that is in the mitochondria, which creates the ATP, and these are sort of short strands, and or for various reasons only inherited from the mother. So this creates sort of a simple family tree. So when we talk about phylogenetics, there will be sort of the mitochondrial tree, and there's sort of an age to the first supposedly mother of all humans. It's not really clear of what that means, but that's sort of what the phylogenetic software can try to date and localize. Okay, so these are all sort of on the small side, and I'm not going to let's go to the other large extreme. So we're the Homo sapiens, we're here at 3 gigabase pairs. And we see that we're not the largest, that there are these fish that have, and plants that have larger genomes, and, you know, plants that we would consider very simple, that are not that far behind. And you can also compare, you know, yeast is quite a bit simpler. So this sort of gives you a sense of the range. So we haven't talked about how much of this is real information. And a lot of the plants that have such huge genomes, they're sort of the same gene repeated many, many times. So that actually makes the sequencing a lot harder. And so having a larger genome doesn't mean that there are more different types of protein necessarily. So this has to be, one has to look further to interpret these numbers. Okay, so this is the process of reverse transcription. So basically the retrovirus invades, it unpacks itself, then the viral RNA turns into this blue copies that are DNA, and then this gets encoded, this gets integrated into the genome. So this is what I described earlier. So this is going to be a useful tool. And a lot of the useful tools is that they're these, you know, so the reverse transcriptase is protein, and it's probably made out of, you know, several, most complicated proteins have several different linear molecules that come together. So you can use this to your own good to change RNA into DNA, for example. But this is also another sort of thing to think about if you're trying to do the, you know, phylogenetic inference. Well, what is the ancestry between species? Well, they could have been infected by the same virus, and part of our genome comes from retroviruses, or so people think. So this is sort of horizontal transfer of information, you know, that violates the sort of tree structure. Okay, I think I already made these points. Okay, so I want to go on a little to some of the engineering. So what can you, you know, do in the laboratory? So at the end of this lecture, we're going to understand, you know, how PCR tests and sequencing is done, you know, because this is sort of a tremendous achievement to be able to make these huge libraries of proteins and, you know, genomes of thousands of people, et cetera. So it's useful to sort of know how it's done. So this is sort of going to the test tube. And I mean, it really is quite simple. So what we want to do is we have a small amount of virus, the RNA virus, just trace amounts. And we want to make many copies. So there's an, you know, almost macroscopic number of copies. So for various reasons, it's easier to work with DNA than with RNA. In you know, theory, maybe you could imagine that you would just use some RNA polymerases and amplify, but the DNA has a lot less mistakes, and that's the technology. So the first step is to, well, let's go to the two-step procedure. To you start with RNA template. So this is your sample. Water and a buffer, you know, keeps the pH at the right value. And then there's the enzyme, the reverse transcriptase. So this would come from a virus. And then these are the DNA nucleotides, the triphosphates. So there are 3 phosphorus chains. So you need to have 4 types of this. And then you put it together at the right temperature, and you get the complementary DNA is created out of this. And then you want to amplify it. So we'll discuss what the primers are, but the replication has to start in it has to get started. So these are sort of short segments that have to match something on the DNA that you have produced. So this is the complementary DNA from this original template. And then the raw materials, so these 4 bases, nucleotides with a triphosphate for the making more DNA, and water and a buffer have to be provided. And then you heat and cool this, and we'll describe what happens, but you get this, if everything goes well, exponentially amplified complementary DNA. And actually, it would have both the complement and the complement of that. So the single strand is used as a template, and you can basically, if you have enough raw materials, create an infinite number of copies. It's basically the reaction stops when you don't have either the concentration is too high or you run out of these base pairs. So what happens here? Well, here there's, these are the 2 strands. And what happens is that when you heat them up to, I think it's 95 degrees, or this DNA polymerase comes from some organism that lives in hot springs, so it can reproduce at high temperatures. And it turns out to be the most useful for this kind of thing. So the first step is the annealing of these primers. So we discussed that the hydrogen bonds are weak, they're kind of marginal. So that sort of means that, well, if you just have a few that match, the thing isn't going to stick together. But the primer is going to be tightly bound if all of these match. So this sort of looks like something that has been replicated. And basically the Taq polymerase doesn't, it always goes, reads from the 3' to the 5' direction. So when you lower the temperature a bit, it'll keep on going in this direction and make a copy on the complementary strand. I mean, that this would, at first it would only, well, you would probably have just one strand, and then you copy in this direction, it stops. Then you heat up and you get the heat causes the hydrogen strands to break. So the double helix gets denatured. And then you can, when you lower the temperature again, first you get these primers to bind, and then there's replication up to this point and replication to this point. So just anything between these two primers gets copied. If everything goes ideally, the number increases by a factor of 2 every single cycle. So it's literally exponentially. But if you don't get the matching of the 2 primers, then nothing happens. So it's a good way of selecting that you selectively amplify. So I just pulled this out of Google. This is not cutting edge lab equipment anymore. This is something that is somewhat expensive, but you know, it's so widely used that it's a commodity item. And other than the reagents and the primers, which you can order or have made for all sorts of things, basically having something where you can do this thermal cycling. You know, it's a thermal cycling between these 2 temperatures. It's just the right rhythm that makes this whole thing work. So that's PCR. Wait, why? Oh. Okay, I think I made all these points except the last one, that if you want to make a PCR test, you, I mean, you want it to be selective. So you want to choose your primers so that it's something that is unique to the coronavirus, then to this particular one, and not any. I mean, you might want to have something more general if you're doing other sorts of research, you know, maybe with bats and other animals. And one way the test could fail is that if you have mutations in the region where the primers are supposed to bind, which would mean that you wouldn't get the amplification. So with most of the tests, there are 3 regions that are targeted and they're chosen. And the other sort of bit of technology with PCR is that if you're looking for a certain sort of feature, you can also put in a third oligomer, so sort of a short sequence of DNA that binds to something in the middle, and then it can have a fluorescent dye that will only become active if it's bound to something. So you can, from the presence of fluorescence, deduce the amount of product that you've made. And this is what is used in so-called real-time or quantitative PCR. So you, at each cycle, you look when the fluorescence starts, and that gives you an idea of, well, really the logarithm of how many pieces of DNA or RNA were present when you started the amplification. The other point is that you know, most biotechnology is it's clever, but it's not terribly creative. So basically, all of these, there are a lot of enzymes and things that nature has developed, you know, like the reverse transcriptase, and we're sort of like viruses, and that with the biochemistry experiments, and that we take these things and then we, you know, purify them, put them in a test tube. But that's much more efficient than, you know, trying to create your own complex molecules from first principles and using them to sort of analyze complicated molecules in biochemistry. Okay, other things that are useful. So, I mean, I think if we would sort of want to make a technology dream list in terms of experimentation, you know, you would like to, you can have all these databases with genomes and proteins, and you would like to just have printers, you know, where you just take some piece of software and you make this and make that and put it together. But for various reasons, there are these, you know, enzymes that can do much of this. So there are these restriction enzymes that can cut DNA based on certain motifs, and then ligases means it can sort of put them together. So here you have these so-called sticky ends where, you know, they match. So it's a little like what we talked about with primers, but just because they match doesn't mean that the backbone will be put together. So you need enzymes to make that happen. But a lot of, you know, biotechnology and research is, you know, these are tools that can be used to do all kinds of things. Okay, so in the last few minutes, I want to talk a little about how sequencing is done, and you know, there's been an evolution in this field. So the first sort of trick is sort of sorting according to size. So electrophoresis means that you create an it's sort of like a type of chromatography. So you have, if you have charged molecules, then having a potential difference sort of tries to drag them along. And it turns out that DNA is negatively charged with pretty much you know, uniform amount of negative charge per unit length. And that's because it's sort of an acid. There are these, you know, phosphate groups, and the hydrogen can dissociate. So this also is pH dependent. So, and then there's a gel, and you know, depending on the length, there's sort of a different rate of being sort of moving through this electric field. So if you put a spot here, the larger segments will move through more slowly, or it'll take them a longer time. So part of it is like a random walk. And then there's also sort of the linear drift, but it's sort of just like the stock market with the stochastic modeling. On the long term, it's always the drift that wins out. So the features, get more defined. So this is gel electrophoresis. So this is the physical principle. And then how can this be used to sequence DNA? So by sequencing, we mean we want the code for the, like I showed you at the end of last lecture, the coronavirus in order. So with the Sanger sequencing, what you do is, so we started a certain place. So this is like in the PCR with a certain primer. So you decide where you want to start. But rather than taking the true nucleotides, there are these sort of defective nucleotides where the OH, this is on the 3 carbon, is missing. So basically, you put a lot of this in, but not that much of this. But this stops the chain. So this is sort of chain termination. And depending on what the ratio is, it'll stop at different points. And then a fluorescent dye molecule can be added to this. So you get from this process, these chains that have been terminated of different length with different fluorescent markers at the end. And then you put this through capillary gel electrophoresis. So it's something like a very thin capillary with about 1 meter of gel. And there's the voltage or current that is sort of trying to drive stuff through, but the heavier, the longer segments come later. And then you have a laser to excite the dye and a detector, and you can basically read out from the chromatograph the sequence. So that this is Fred Sanger won 2 Nobel Prizes. One was for this, and the other was for sequencing the first protein, insulin. And I believe it was the late 1950s. So this is, works well, but it has a number of drawbacks, and that it's very tedious, and there's sort of a limit to the length that you can deal with this. So it would be very expensive to do sequencing this way. I mean, the Human Genome Project, if you read about its history, it was sort of this big project, and then with new technology, much of the same can be done at now almost negligible cost. So what is better? Well, if you talk to people in computer science, their buzzword is massively parallel and scalable. I mean, there are 2 bits of technology. The first is sort of optical, and that you want to be able to sequence a huge number of short sequences that are sort of all different. So you want to make an array, so you can sequence simultaneously like a few 100,000 different sequences. So you have this array, and it's sort of almost at the diffraction limit. And what you do is you break up the DNA in random sequences. They shouldn't be too long, but not too short. And then they're attached to these chips, and they're made to, they're multiplied, so you have these bunches that are the same. And you attach the bases with fluorescent markers, and basically there are different turns. You go, you have fluid having just one and just the other. I mean, you loop through the 4 bases, and then you image and you see which of these pixels light up. So which strand has just added something. So basically there's sort of a beat or a loop where you go through these different bases that you add, and then you record, you know, the strength of the fluorescent signal. So there are errors to this, and the segments can't be too long, because eventually it sort of degenerates down into noise. That you have sort of many badly sequenced or somewhat badly sequenced bits of genome, and then you, with a computer, you put it all together. So this is a lot faster, a lot less expensive. There's some drawbacks to it where you have features that repeat that are longer than the size that is sort of order 500 base pairs that you can sequence here. So there are limitations with this method. There are other, this is next generation. There's also 3rd generation where you have these micropores, and you can follow the current, and from the current you can see each base go by and what the type is, and that can sequence longer segments than this. But this is basically, you know, how it is done with, you know, for the coronavirus and medicine, you know, for sequencing, and 90% is now by this technique. Okay, so today has been very sort of biological, technological, because I think there's a lot of jargon and sort of background material. So I think I've gone through, you know, most of that, so you can sort of have an idea of how it's done. And on Wednesday, I want to go more toward theory. So I'm going to talk about mutations. I mean, perfect copying, or to the extent that it's perfect, is really quite boring. So I'll talk about models for predicting the likelihood of mutations. I'll talk about genetic clocks. I'll talk about mathematical models of selection and genetic The jargon is drift, but it should be diffusion. This is random walk, whereas natural selection is sort of being pushed in many cases very slowly toward, certain improvements in the genome. Then I would like to talk about phylogeny. So that is sort of how to make trees, be it for the coronavirus or for all of life. Comparison of genes between species in bioinformatics. So the next two lectures, we'll talk about the coronavirus and the new strains and the proteins and do that in some detail. Probably in the last lecture, but in the next lecture, I sort of want to focus a little more on the theory, where we sort of take some of the results of this sequencing and experiments, and we try to make sense of it and use it for different purposes. So thank you for your attention, and I'm ready for questions.
Yeah, Martin, thank you very much. It was really an interesting and very informative lecture today. Thank you so much. I'm sure there are some questions, and please unmute yourself and ask. Or use the chat if you want to avoid using the audio. And Martin, I think for Wednesday, I need to change the Zoom links and give you 3 hours. So remember, there is a voucher to be won.
So we can't ask ourselves questions.
Yeah, if you want a voucher, Martin, you should ask a question now. Maybe you can allow Ilya to ask question for a voucher, because he's only also a participant today. Martin, it's
sorry, Martin.
Here we come. So, book voucher for Ilya.
I don't need the voucher. I don't need the voucher, but I can still ask a question.
Yes, please.
Sorry, Martin. And do you know, like you were talking a lot about this PCR test and how we can kind of make sure that this PCR test works fine, that we kind of looking for a specific part of the genome and so on and so forth. But what about the problem with the PCR test, that they take time. And there are some other tests with respect to the coronavirus, for example, like lateral flow tests. And do you know anything about those tests and why they're not so precise?
I really haven't looked into it.
Okay. No, I just maybe. Okay, sorry.
I mean, from my understanding, the antibody tests are based on, if I could sort of maybe add a sort of general idea, this idea of sort of matching patterns, so I mentioned the primers matching. So you have antibodies and antigens and stuff sticking to each other. That is a very common sort of test. another sort of test, I didn't want to, go into a long discussion of biotechnology, but sequencing isn't the only way. So there are, libraries of patterns. So for example, there are, I think, a few 100,000 different, single point differences in the human genome. So you can make these sort of chips where stuff sticks with a fluorescent dye, and then you sort of use these chips with, where does it light up to classify differences, where there are sort of locations and different choices. So that's done for a lot of the medical tests. And these Illumina machines, I mentioned that a lot of it is an optical system almost at the diffraction limit. And yet a lot of those machines can also take these kinds of chips. So that this is sort of other technology that what one can use. But I haven't really, there's just so much to read. I can give you a good answer to the question.
There is a question to read in the chat. If you would like to comment on that.
Can you read it to me? Because
the question by Teto is, how is it that COVID-19 vaccine have been developed in approximately 1 year, but viruses take much longer to mutate? Don't vaccines get outdated very quickly?
Well, that's a good question. The mutations are the coronavirus is 30,000 bases long. I guess since it's single strand, there's no pairing, but that's a detail. And you get about one mutation every 3 weeks, very roughly. So they're going to be sort of, at most of the sites, very few, at any site, there's a sort of 1 out of 1,000 chance of there being a mutation. And most of the mutations, so there are just tons of mutations that have taken place, but most of them don't matter. And most of these vaccines are very targeted, so they'll just target a certain area. So what we saw was, Tulio de Oliveira and others in South Africa have been working for a long time sequencing the coronavirus. And to be somewhat frank, I think not everyone appreciated that this is important. And it was sort of, oh, well, this is just people doing stuff off in their lab, but, and then, of course, the news strains came along that are, have very different properties. So these are mutations that matter and that make the virus, either spread a lot faster, evade vaccines, become more virulent, but as we will discuss in the next lecture, structure. Most mutations are either really deadly for the virus, so we don't see them, or they don't matter at all, and it's just these sort of few events that you know, everyone talks about, and it's really hard to predict. So, you know, we look at the influenza virus. It's sort of moderately successful, and it has to be updated every year. We look at sort of the classic vaccines that everyone gets in their childhood, and those prove to be robust and not go out of date. With the HIV virus, from what I understand, the vaccines that you find a target and then the virus mutates to get around it. So it's a good question, and I don't know whether I don't think I've given you a very good answer to it, but that's sort of the uncertain answer.
But at least we have a winner for the question competition.
Yes.
Congratulations. Teto, please send me your email address so that we can sort out the formalities. Martin, I think it's time to come slowly to an end, also because in 20 minutes we have the next talk, the NITEP colloquium, where everybody is, of course, kindly invited again to join us. Yeah, in particular, people that attended Ilya's lecture should come because it's closely related to that. But Martin, you're invited as well. Yeah. Okay. Wonderful. It was a busy day again today, and I hope we all learned a lot. And we meet again within the school tomorrow morning. And I have to confess, I don't remember if it is Morten or Charles tomorrow morning, but it probably doesn't really. It's Charles tomorrow morning. Yeah, the first speaker is Charles Crosby tomorrow morning, and then in the afternoon we are back to machine learning. So thank you very much for being with us again. And I hope to see you again all at 4 or latest tomorrow morning at 9 again. Thank you very much. Have a good evening. And Martin, thank you very much. It was really a nice lecture. Thank you very much. I enjoyed it very much.
Well, thank you. Bye.
Thank you. Bye. Bye-bye.